And our next speaker, Pingze Zhang, also from MPI. Let me put up the slides. Just 1 sec.
So first, many thanks to the ARDD committee for giving me such great opportunity to present our story here. So I'm going to talk about how does S6K influence regulate lifespan and potentially also conserved in the human and the mammal systems. So we know that rapamycin is a TOC1-specific inhibitor and has been shown to has been proved by FDA for many clinical diseases, including renal transplantation. In addition, rapamycin treatment can increase lifespan in multiple model organisms, ranging from yeast to mice. And one recent clinical study conducted by John Monick could show that TOC1-specific inhibition by using rapalog can improve the immune function in the elderly. Therefore, as one of the most promising anti-aging drugs, understanding the downstream mechanism of rapamycin during the aging process is essentially important to apply this drug to benefit our human longevity. We know that rapamycin-related lifespan extension requires two key components. One is autophagy and another is S6K. And one recent study from our lab could show that gut is the essential organ to mediate the autophagy-related lifespan benefit. However, for S6K, although the S6K mutant mice show a longer lifespan compared to the control group, but it's still largely unknown how does this protein regulate the aging process and which tissue mediates this benefit. Therefore, we can explore this project using the Drosophila model as a model organism to answer this question. We first established that knocking down S6K activity ubiquitously in flies is beneficial for lifespan. In addition, we further screen which tissue are important for this lifespan benefit. We therefore knocking down S6K activity in multiple fly tissue to see which tissue is essential. However, we noticed that it's not about brain, muscle, heart, and gut. None of these tissues show lifespan benefit. But in the fly fat body, we could see that there's already lifespan extension upon the S6K knocking down in this tissue. In addition, we could also show that this lifespan benefit regarding to S6K in these fatty bodies are also on the downstream of the rapamycin treatment. You can see upon the overexpression of the S6K activation in the fat body cells can strongly attenuate rapamycin-related lifespan extension. Then we ask another question. Why does this protein in fat body are so important for the aging process? Therefore, we conducted proteomics analyzing to answer this question. We noticed that there's many lysosome-related annotations are strongly upregulated upon the rapamycin treatment or S6K knocking down in the fat body cells. We therefore further verify this finding through the lysosomal staining in the fat body cells. And interestingly, we notice that there's a lot of enlarged lysosomes indicated here upon the S6K activation and together with rapamycin treatment in the fat body cells. And the quantification are on the right panel. In addition, we further confirm these findings through the electron microscopy. We notice that these enlarged lysosomes are not only enlarged, but also show a multilaminar structure in these lysosomes. Then we ask another question. What caused this lysosome morphological change? And we know that lysosome is the endpoint for lysosome autophagy pathway and also essential for the endocytosis pathway. And we also know that the vital role of the autophagy during the aging process. We therefore first evaluate whether autophagy can explain these lysosome changes. Therefore, we inhibit autophagy by overexpress ATG5 knocking down strands in the fat body cells. And however, we notice that the S6K activation induced enlarged puncta cannot be affected by autophagy inhibition in the fat body cells, which suggested that at least this lysosome morphological change upon S6K manipulation is not regarding to autophagy pathway, or at least not regarding to the ATG5 dependent autophagy pathway. However, how about endosomes? We notice that upon impaired late endosome in the fat body cells, there's also similar enlarged lysosomes in these fat body cells, which means that probably the endocytosis pathway is essential for this lysosome morphological changes. Then we ask another question. Why does S6K and the mTOR signaling can regulate these lysosome changes? What's the molecular link between this process? Therefore, we go back to the proteomics analyzing, and one of the candidates caught our attention is Syntaxin 13. This protein is highly regulated upon rapamycin treatment or S6K knocking down in the fat body cells. In addition, this protein is a Syntaxin family protein which mediates the fusion of the vesicles, especially for the endosomes. We therefore ask that whether it's regarding to the lysosome fusion stage. And interestingly, from the human cell culture study, we've noticed that it's already published by the other group. Syntaxin 13 knocking down in these human cells also induces the multilaminar and enlarged lysosome in these cells, which suggests that probably it's an explanation for our phenotype. We therefore knocking down syntaxin 13 in the flat fat body, and interestingly and consistent with our hypothesis, we notice that there's also enlarged lysosomes in these fat body cells. We therefore further verify these findings through the epistasis study. Here you can see that the S6K activation induced enlarged puncta can be strongly attenuated by syntaxin 13 co-expression in the fat body cells, which means that syntaxin 13, this protein, is on the downstream on the TOC1 S6K signaling to mediate this lysosome enlargement. How about multilaminar lysosomes is still unclear. And we further confirm that it's not only about size, it's also about the structure of these lysosomes. Here you can see that the S6K dependent multilaminar lysosomes are also strongly attenuated by syntaxin 13 co-expression in the fat body cells, which means that syntaxin 13 is under control of the TOC1 S6K signaling to mediate the lysosome morphological changes. Then we ask another question. Because we notice that S6K is essential for longevity and aging process, but whether this lysosome morphological change is also essential for the aging process is still unclear. Whether there's some physiological meaning for this process, it needs to be further explored. We therefore go back to the proteomics analyzing to see what happened in the old fat bodies upon rapamycin treatment and S6K knocking down. And interestingly, we notice that in the old fat bodies upon rapamycin treatment or S6K knocking down, there's many immune-related annotations are strongly downregulated, which probably stand for the beneficial effect for the immune functions. We also observe that during the aging process in the fat bodies, there's a activation of the NF-κB-like transcription factor relish, and this protein will go into a nucleus to stand for the inflammatory aging conditions. We therefore ask the question that whether TOC1, S6K, and syntaxin 13, this signaling can mediate this age-associated inflammation in the flat fat body cells. We therefore conduct a relish staining for the fat body cells in the old flies, and we notice that rapamycin treatment can strongly attenuate the age-associated inflammation in these fat body cells. And this phenotype is dependent on S6K. You can see S6K activation completely rescue this effect. In addition, we could further confirm that it's not only S6K, but also the syntaxin 13, the candidate we identified before. So here you can see syntaxin 13 or expression can strongly attenuate S6K activation, rescue the effect on the inflammation condition in these old fat body cells. Therefore, we know that the TOC1, S6K, and the syntaxin 13 signaling in the Drosophila fat bodies are essential for this inflammatory aging condition during the aging process. So then we ask another question, whether this age-associated inflammation is detrimental for the aging or it has other functions with other age-associated dysfunctions. Therefore, we first explore that whether the inflammatory aging conditions in the fat body cells are essential for the age-associated immune functional decline. Here, we use the bacterial infection assay to answer this question. So we first knocking down relish specifically in the flat fat body to mimic a ameliorating status of the inflammatory aging. Then, at the old stage, we infect these flies with bacteria and to see how about their immune function to clear this bacterial infection. And interestingly, we notice that upon the inhibition of the inflammatory aging in these fat body cells, these old flies show a better immune function indicated by improved bacterial clearance rate in these flies. In addition, we could further show that these flies show a longer lifespan compared to the control flies. Based on these findings, we could say that the inflammatory aging conditions in the flat fat body is detrimental for the immunosenescence and also essential for longevity. We therefore ask another question, whether our findings in flies are also conserved in the other model organisms and maybe also for mammalian systems as well. Interestingly, from the publication, we noticed that no matter rapamycin treatment or S6K mutation, in yeast and worms, there's also elevated syntaxin 13 ortholog levels. Then we ask, how about mice? Therefore, we use the mouse liver to answer this question. The reason why we choose liver, because liver is a functional equivalent tissue to the flat fat body. And we notice that upon rapamycin treatment, this liver show elevated syntaxin 13 levels indicated by the Western blot assay here, which means that syntaxin 13 regulation machinery is highly conserved across multiple model organisms. How about the immune part? Whether the age-associated immune dysfunction are also conserved between flies and mammals. Therefore, we conducted proteomics in the old liver upon rapamycin treatment and combine this dataset together with the published rapamycin and S6K mutant transcriptome dataset to answer this question. And we noticed that in all three datasets, there's many immune-related annotations are strongly downregulated upon rapamycin treatment or S6K knocking down, which means that probably it's not only Syntaxin 13 expression levels, but also the age-associated immune process are strongly affected by the TOC1 S6K signaling in both flies and mammals. Therefore, we go to the summary slides of our findings. Here, we could identify that the TOC1 S6K signaling related lifespan extension rely on fly fat body, and this signaling in the fly fat body can mediate the expression of the Syntaxin 13, thus important for the fusion of the lysosome and the late endosomes, which is essential for the age-associated inflammation. And this inflammation condition is detrimental for the age-associated immune functional decline, and which stands for the immune senescence, and also detrimental for the longevity itself. And finally, we could confirm that our finding regarding to the TOC1 and S6K signaling on the Syntaxin expression and the immune processes are highly conserved across model organisms. And due to the time limitation, we only show part of our results here, but our paper already on bioarchive now. If you have any questions on our story, please come back to me, or you can read our paper on bioarchive. And at last, I want to thank all of the members listening here, especially for Linda, Sebastian, and James. And my last sentence. I'm approaching finish my PhD study here, and I'm looking forward to see any potential postdoc position to let me stay in the aging and the immunology part. Thank you very much.
Thank you so much, Pingze. Do we have any questions? Maybe I can start. So I think endosomes can also impact fatty acid metabolism.
Yeah.
Could there be anything in that direction?
That's a really interesting question. So we answer these questions through the starvation assays. We notice that we know that rapamycin can increase the starvation resistance in flies and other model organisms, but this resistance effect cannot be affected by S6K activation. In addition, we could also show S6K knocking down in the fly fat body has no effect with the starvation resistance. We cannot exclude the other part of the fatty acid metabolism, but at least we know that globally it's not changed too much.
Okay, very interesting. Do we have any more? We have one question down there. Yeah.
Great talk. So when you knock out S6 kinase in the fat body cells, does it affect any other organs? I mean, did you check any other organs like brain during the aging study, in the aging study? And if you did, what regulates?
Yeah, that's a really good question. So we are curious that whether there's crosstalk between the fat body to brain and to gut, but we are working on it. We cannot give you too much data about it. Sorry now.
Other questions? I think we have. No, we are actually on time. We will give out another warm hand. Thank you. Great.